Ever wondered how potential drug candidates are identified? The answer lies in a computational technique known as molecular docking. This technique predicts the preferred orientation of one molecule, the ligand, when bound to another molecule, the receptor, to form a stable complex. The key to this process? Scoring functions. Scoring functions play a crucial role in evaluating and ranking the potential binding poses generated by molecular docking algorithms. They estimate the binding affinity between the ligand and receptor, helping researchers identify promising drug candidates. But not all scoring functions are created equal. There are several types, each with its unique approach and application. First on the list are force field-based scoring functions. These utilize molecular mechanics force fields to estimate the intermolecular interactions. They consider parameters such as van der Waals forces, electrostatic interactions, hydrogen bonding, and solvation effects. An example of this type is AutoDock, a popular choice among researchers. Next up are empirical scoring functions. Rather than using force fields, these functions rely on statistical analysis of experimental binding data. They are less detailed but are computationally less demanding, making them useful for high-throughput virtual screening. ChemScore and XScore are examples of empirical scoring functions. Then there are knowledge-based scoring functions. These functions use statistical analyses of known protein-ligand complexes to derive scoring terms. They rely on databases of experimentally determined structures to identify common interaction patterns. Examples include DrugScore and PMF. The fourth type is consensus scoring. This approach combines multiple scoring functions to enhance the reliability of predictions. It helps reduce the impact of individual scoring function limitations and increases the robustness of virtual screening. DSX and CDocker are examples of consensus scoring. Lastly, we have machine learning-based scoring functions. These utilize machine learning models trained on large datasets of protein-ligand complexes. They can capture complex nonlinear relationships between molecular features and binding affinities, such as RF score and NN score. The choice of a scoring function depends on the specific goals of the docking study, the computational resources available, and the characteristics of the system being studied. Often, a combination of scoring functions or an ensemble approach is used to enhance the reliability of predictions in molecular docking studies. In summary, molecular docking and scoring functions are vital tools in drug discovery and design. From force field-based to machine learning-based scoring functions, each type has its strengths and applications. By understanding these different types, researchers can make informed decisions about which scoring functions to use in their studies, bringing us one step closer to the discovery of new, life-saving drugs.